So, class, I need your cooperation if I forgot to record our meeting. So, please kindly remind me. So, before the beginning of the class, kasi minsan ay siguro nakakalimutan ko ang pag-record. So, I appreciate that you will remind me for the recording so that your other classmates can have an access of the recording lecture afterwards. Okay, thank you so much. So I will now go back to my slide. Okay, so for the question again, first question is, it is the technology that uses living systems to produce compounds, drugs, and food. So what is it? Is it A, bioprocessing, B, fermentation, or letter C, recombinant? Okay, so let's proceed to another question. So jot down your answer in your paper. So number two, what products that has been produced during the pre-1900s? So what products that has been or that have been produced during the pre-1900s? Is it A, kimchi, B, alcohol, C, acetone, or D, vinegar? Okay, so the choices are kimchi, alcohol, acetone, or vinegar. So, third question. Okay. Third question is, the fermentation comes from the Latin verb. Is it A, to boil, letter B, to bubble, or letter C, to make? So the fermentation comes from the Latin verb, which means to boil, B, to bubble, C, or to make. Okay, you have answers, jot down it in your paper. And then the fourth one question is, the wave of fermentation industry is the beginning of the pure culture technique. So if you are already familiar, so you can have a wild guess naman. So this wave of fermentation industry, actually it's an event, the chronological event. So tinatawag siyang wave. There are four waves sa history ng fermentation. Is it in the first wave, B in the third wave, or C on the second wave? So just shut down your answers. Okay, bilang ha. Okay, the last question is give or choose the examples of microbial products. It could be sugar, it could be amylase, it could be insulin, but you can answer pwede din all, ganon. So you can there are more than one answers naman dito. So, it could be A, it could be B, it could be C, or it could be BC, it could be A or B or C. A, B, C, ganon. So, yeah, that's it. So, tapos na kayo mag-jot down ng answers niyo, class.
Yes, ma'am.
Okay, so let's now proceed to the lecture. So the lesson one is the introduction to bioprocessing. So at the end of this lecture, you will be able to define the technical terms related to bioprocessing, enumerate the different kinds of microbial products, and then the last one is to understand the relevance and importance of bioprocessing to biotechnology. So to define bioprocessing, so I think you are already familiar with this and you have already siguro understood by heart because as a biotechnologist, you have to, even though you are naka-close yung eyes, you really have that no, concept of what is really bioprocessing or biotechnology. So as a definition, bioprocessing uses living systems, okay, biological systems. Examples of these are microorganisms and plants to produce compounds, drugs, and chemicals that are useful to consumers. So the end users of these are the society or that are really beneficial to the society or to the consumers. So it must be these compounds and drugs have a significant impact. Okay, it could be a protein that can be used for those who are suffering with an ailment or diseases. Yeah, so those have genetic disorders. It could be like that. So yun. So yun ang end product ng bioprocessing. So it is technically carried out through fermentation, and one of the products from fermentation are the commercially available insulin produced by recombinant microorganisms. So the production of insulin in a microorganism is really one of the breakthroughs of biotechnology. So because of this, it has been already has the insulin product is already accessible to any parts of the world, and also it is already available in the municipalities, in the provinces, due to the increasing rise of the diabetic patients nowadays, because diabetes can be genetically inherited, or it can also be it's an onset then. So there are many types of diabetes, right? So ayun, so the diabetes, I think type 1 is needs a maintenance for insulin insulin injection, and because I think that if you have type 1 diabetes, you don't, the body cannot produce insulin in order to regulate your blood sugar. So that is why you need to have a maintenance of insulin that should be shot, that should be injected, through the veins. So, ayun, and then with this kind of rising patients of these ailments or diseases called disease ganon. So yeah, so insulin is very in demand, and you know that class insulin originally insulin are really produced from the are extracted from those mammals or cows ganon, so that they will really extract it and then purify it and then process it to be commercially available. Now, imagine class if really because of the rising demand of these insulin products, so kapila magsige og kanang patay og baka aron ma-extract ang insulin. So it is really costly, and it is really it is a complicated process. So whereas when we really produce it in a microorganism, so they exploit the microorganism, which is in a minute form, which is really very small, that really to the naked eye, ganun. So, but really, it really does multiply in a exponential manner, and it can really produce high amounts of insulin. So that is why they really took advantage that benefit or concept that. So that is why bioprocessing is really a helpful tool to the society, and thus the fermentation is the backbone of biotechnology in the industrial process. So it is an upscale, industrial process meaning an upscale na talaga. There is a large scale production of this product. So that is why it is one of the breakthroughs of biotechnology, so the insulin product. Okay, so you will all the more know about the methods of recombination or the methods of recombinant DNA technology when you take InBt134, but I guess you have already an idea what are these processes and techniques in order to clone a gene in a microorganisms, ganon. So yeah, but before we'll focus on these ones. So what another term is that industrial biotechnology. Okay, so it is the exploitation of enzymes and microorganisms and plants that produce energy, industrial chemicals, and consumer goods. So it is a broad area in biotechnology, and bioprocessing is under of this industrial biotechnology. So it is an industrial paradigm based on the expectation that renewable plant-derived carbohydrates, lipids, and other compounds can displace a significant fraction of petroleum and other fossil fuels that are currently the raw material and energy basis of modern industrial societies. So it could be in the industrial biotechnology, it is not just about medicine, ganon, but also it can also be of help to fuels and petroleums that are really that are also in demand, that are used by the society from day-to-day basis. So they use renewable plant-derived carbohydrates in order to produce high amounts of petroleum and other fuels, no? So in order to have a combustion in their mga cars and other transportations. So that's one of the area of industrial biotechnology. And another area then also in the medicine that it can also have a large scale production for those enzymes and proteins that are really useful to the society. So now the objective of this industrial biotechnology is to develop biotechnological approaches or methods or techniques, processes that can yield green industrial processes that are cost effective and sustainable. It should be green. What do you mean by green? It should be environmental friendly. So when these products are produced, they cannot harm the environment, and when these products are discharged or thrown into the environment, they will not abrupt the food chain systems or the food web in the ecological environment, ganon. And then second, it is cost effective. It does not require such high amounts of yung mga high amounts of money in order for that process to be realized, no? So it will result to affordability of the product, and then it should be sustainable. Yun naman talaga ang goal ngayon in every project, that it should be sustainable, that it will really be it could really be applicable in the next 5 years, in the next 3 years, in the next 10 years, ganon. So, and then also it is cost effective, na it does not take a lot of resources in order for that to carry out. So that's really the main objective of the industrial biotechnology. Okay, understand class? So let us proceed to the specific terms about fermentation. Okay, so specifically, fermentation is really the backbone of the bioprocessing, and the term fermentation comes from the Latin verb fervor, okay, meaning it is to boil, because they have observed from the sa atong mga ancestors from the past ng mga people, the past ancestors that we have, because they have observed that the action of the yeast, the action of the yeast, it produced bubbles or kanang mga, it really just like the water that is boiling. So yun siyang bubbles, ganun. So meaning fervor means to boil. So they observed it in the yeast, the fermented fruit acted by the yeast, or the molded grain, so acted by the yeast. So to further define fermentation, so it is the generation of energy by the catabolism of organic compounds, whereas its meaning in industrial biotechnology or microbiology tends to be much broader. Okay, so mamaya let us define technically the fermentation process. So the first industrial process rather, sorry, is the alcohol production using yeast. Okay, so one of the products of the fermentation is alcohol that is acted by the yeast. So technically, it is a metabolic process that produces chemical changes in organic substrates through the action of enzymes. So there is no chemical reaction involved because the enzymes are the ones that are really taking place for that in order to produce that particular product by the microorganisms. So in microorganisms, it is the primary means of producing adenosine triphosphate. So this is the energy. So ATP is the specific energy that is needed by the microorganisms in order for them to grow. So through the degradation of the organic nutrients anaerobically, or could be aerobically then. So you have to feed your microorganisms in order for that to grow. So it produces ATP. So and then the amount of the food or the medium that is grown by the microorganisms, it will be depleted, but then the microorganisms will multiply exponentially. So that's the role of fermentation, or the technical process of fermentation in microorganisms. So the equation word for fermentation is when you have a sugar or the glucose, so it will be acted by the microorganisms, by the enzymes of the microorganisms. For example, yeast, it will produce ethanol, then carbon dioxide, and it also have sugars and other sugars, and then it is a byproduct of carbon dioxide, which is in the form of bubbles. That is why they observe it na nag-boil itong sa ating mga ancestors. That's why they have named fermentation to boil. Ganun. So that's the presence of carbon dioxide that are released from the fermentation medium. So that is why yun ang kanilang observations. They don't know yet the processes, they don't know yet the mechanism of this fermentation. Until then, the scientists and the curiosity of the scientists are renowned scientists know they really discover, and they really wanted to have an explanation of all these things. Okay, so that's it for the equation for fermentation. So let us now proceed for the 4 groups of microbial products. So we will discuss this one by one. Okay, the microbial cells, which is the biomass, and then another products are the microbial enzymes. There are many products of enzymes by the microorganisms. You can also have microbial metabolites. So, and the 4th one are the recombinant products. So transformation processes are really the techniques or the what they call the process flow in order to derive a such recombinant products. Okay, so okay for the microbial biomass. So those when you really wanted to ferment. For example, yeast. Okay, you can really use the yeast after the fermentation. You can. They also use the alcohol product because you have the yeast can produce alcohol, and you can also use the biomass of the baker's yeast. So to be have a single cell protein that is used as human or animal food. So it could be used as feeds for the animals such as pigs, at the goats, chicken, ganon. So yun ang mga examples that these biomasses are rich in proteins that can be used as a human food or animal food. So in the early 1900s, when the First World War broke out, so the baker's yeast has been produced on a large scale size and large scale amounts. So it was used as a human food because there are really scarcity of that food because of the World War that was happening that time. So that is why they have to produce such food that are really high in protein in order to have that strength, you know, so you can really acquire such strength from the protein so that you will really survive for that war, you know. So that's why they used baker's yeast and then as a human food for the during that time. Okay, so that's one of the example of the microbial biomass. Okay, so another silver example is when I became a research assistant in field crops. So we really our project intended to make a microbial inoculant or microbial starter in a wine, particular wine, which is taro wine. So because there are many starters naman that are available in many places, but then was a demand that it should be produced in a sterilized condition and also in an optimized manner. So that is why this the cells or the microorganisms that are present from those traditional microbial starters were isolated. Then these those microorganisms that played such significant role for the wine fermentation, it was really then produced in a large scale. So marami yung involved ang yeast because it produces alcohol, it produces esters, those aromatic compounds that we can really smell from the wine. So those are from the yeast, the Paicia specifically. So yun, mga ester compounds yun. So that's why those microorganisms are produced in a large scale, and then those microbial biomass are taken out, and then it will be processed into a microbial starter, and it will bind in a flour. Ganun. So that's one of the uses of the microbial biomasses for the wine fermentation. Okay, so that's just an overview of our project way back 2013 to 2016. So that's it. So in the 1970s, it became popular as a source of protein for animal feeds, and there was a large continuous process that was carried out. So baker's yeast is popular nowadays because of this as used for the leavening ingredient of bakery products. So there are many bakery stores nowadays, and there are many who make bread. And even in the time of the pandemic, there is a high demand of these bakery products. So that's why baker's yeast are really not phased out in the market. It's still continuously being reduced. So kaya marami and then also there are many households that are also using baker's yeast in order to ferment such fruits. No? So they just have to make a homemade wine. Pineapple wine ba yun? Mga fruit wine. So ganun. So it is really important and significant in our present generation. Okay? So next naman are the microbial enzymes. Of course, microbial enzymes play a very significant role. So the production of enzymes have been carried out commercially from different plant, animal, and microbial sources. So you can extract the enzymes in the plant. You can also extract also from animals, but you can really produce high amounts of enzymes from the microorganisms. But yeah, and then the enzyme production is closely monitored in microorganisms. So, but in this production of enzyme, there is still a critical conditions that has been that should be monitored or closely observed in terms of pH, temperature, substrate concentration, fermentation incubation, and so on and so forth. And it should be optimized in order to produce an active enzyme or a not denatured enzyme, because there are also enzymes that are not really that are denatured during the fermentation process because of mishandling, because of temperature sensitivity of the enzymes. So ganon. So it's really crucial when you opt to do your thesis about enzymes. It's really, I know, critical that you have to observe such conditions such as pH, temperature, and many other things. Okay, so substrate concentration, so ganon, agitation during the fermentation, so ganon. So these are the enzymes, that are really very sensitive to such conditions. So the examples of microbial enzymes that can be found in the microorganisms, these are amylases, proteases, pectinases, cellulases, beta-glucanase, beta-glucosidase, dextranase, invertase, lactase, and glucoamylase. Diba ang enzymes, these are the biological products that can hasten a reaction without without the presence of such chemicals. No, so these are the things talaga that it can also degrade carbohydrates, ganon. So these amylases are also present in our bodies because it will degrade glucose, it will degrade the proteases, degrades proteins, ganon. So pectinases degrade also pectins, and pectinases are used in the yung mga pulp juices, mga Minute Maid, ganon, because there are many pectins in that in the fruit juices, so it should be degraded. That's why they have to add pectins to it so that it will not be sold na pulpy siya, ganun. Pero meron ng iba na pulpy na yung ibang mga juices right now. But then, yeah, kung may mga pulp siya, kung wala na siya pectin, in order to have a homogeneous liquid form of your juices in the processing of these juices like calamansi juices, pineapple juices, ganon. So that's it, pectins. So there are many more uses of enzymes that are really have a significant impact in the food industry, in the medicine, in the hospitals, ganon. And also in the Philippines, microbial net. So if you are familiar with that, um, my one of my study leaders actually in that microbial starter project. So one of the study leaders, which is the pioneering, also project leader, which was also my professor, also in MBB 240 during my master's study. So she was the pioneering scientist to produce microbial rennet that is an alternative enzyme in order to produce to make cheese. Okay, so these the microbial rennet is the enzyme that is responsible for the clotting of your milk in order to separate the proteins, okay, from the milk, ganon. So this is the alternative product because rennet has to be extracted from a mammal. So, ganun din yung concept niya sa insulin. So, if that's the case, you have to, of course, the microbial rennet is available already commercially. Ganon. The rennet is already available, but it is really expensive when these products are really came from these animals or mammals because of the many processes that are involved. So that is why they, Mom Susana Mercado, or Professor Susana Mercado, paved the way for this discovery of the microbial rennet that is produced from the Rhizopus chinensis, which is a bread mold. Okay, until now, they are really got to sustain these microorganisms in the lab, and they reproduce microbial rennet. And I have also during Christmas season, so I always buy these cheese that are available in the lab. So kaya meron na din cheese industry that who adapted the technology. Kaya ang ganda talaga ng ano nila product. So that's how this microbial rennet paved the way for the cheese production. Okay, so another microbial product is the microbial metabolites. So there are two kinds of metabolites produced by the microorganisms. First is the primary metabolites, which is these are amino acids, nucleotides, proteins, and nucleic acids, could be lipids and carbohydrates. Okay, these are produced during the log phase, which are essential to the growth of the microorganisms. So, in other words, the primary metabolites are produced because it will be used as a it is used in the growth of the microorganisms for the multiplication or the proliferation of the microorganisms. So yun, so these are really useful and beneficial to the microorganisms. So that's why they are called primary metabolites. So some of these enzymes are one of the primary metabolites that are useful in their proliferation because sometimes the enzymes, most of the time the enzymes degrade these glucose or these sugars in order for the microorganisms, for that glucose to be broken down in small pieces, and this glucose shall be the ano ba yun, yung kanilang in the metabolic, these are needed in their metabolic pathways in order to produce ATP or energy so that they can proliferate. So yun ang use ng enzymes. So it belongs to the primary metabolites. Okay, class, are you still there?
Yes, ma'am.
Okay, so let's continue. So I guess we will not ano, sige lang kung asa kutub until 11 a.m. Then another one is the secondary metabolites that are produced during the stationary phase. So, yung log phase class, later we will talk about the microbial growth kinetics of the microorganism. The log phase class, it is the exponential growth of the microorganisms, meaning the microorganisms are actively dividing, ganon. So, it's really a through binary fission naman yung ano ng microorganism. So, it is really actively dividing. And then, there is a fast fast degradation of the nutrients in the media of that. So, while in the stationary phase class, no. So the stationary phase is there is now the depletion of the nutrients, and then also the microorganisms are not actively dividing, but they are not yet no, hindi pa sila into the cell death. But there is really now a stability of their growth that they will not proliferating that much. Okay, they're still dividing, but they're really not in the exponential form. So that is paving the way to cell death because there's now a decrease or a small amount of the nutrients that are because the more, di ba, microorganisms mo proliferate, so mudaghan ang cells, ganon. So the cells are really proliferating. So they are have a inhibition or competition of the nutrients. That one. That is why when there are no nutrients, so it will pave the way to cell death. Ganon. So that's the stationary phase that the cells are still are alive or are really viable, but then there's no available nutrients that can be used for proliferation. Okay, so and then these secondary metabolites are produced now. So it could be that these secondary metabolites are that has antimicrobial activity, which can be used as a defense mechanism of these microorganisms from the other compounds or other microorganisms also that are present in their niche, for example, in the environment, or other specific enzyme inhibitors. Some are growth promoters also, and many have pharmacological properties. So these are mga secondary metabolites na that have antimicrobial activity in order to so that the microorganisms will thrive pa longer, ganon. So because they do not have a nutrients also that are available in the medium or in the environment that they are in, the nitrogen that they are in. So they have to find ways. So these are the secondary metabolites that are produced that has microbial activity, antimicrobial activity. So ayun, and these antimicrobial activity these are antibiotics, di ba? Kala mga penicillin, mga cefalexin, mga erythromycin. These are secondary metabolites that are present that are produced by the microorganisms. So these are really exploited so that they can really also kill other microorganisms. So this is used as a defense mechanism from other microorganisms also. So ayun, that's the role of the secondary metabolites. Could be used as hindi na siya ginagamit for growth of the microorganisms because it is produced in the stationary phase and then it is not really actively dividing na din yung microorganisms. It is just for the sustainability or for the microorganisms to thrive. So before the cell death comes, the cell death will pave the way for it, you know. So that's the use of the secondary metabolites. Okay. And the uses could be growth promoters and also have pharmacological properties such as antibiotics. So yun ang mga important, significant uses of these secondary metabolites. Okay, so How about the recombinant products? So these are the examples of these are the interferon, insulin, human sebum, albumin, epidermal growth factor or calcinosine and bovine somatostatin. And the common host of these products are Escherichia coli or E. coli, and also we will have also the yeast, the Saccharomyces cerevisiae. These are the mostly commonly investigated host cells for the expression of these recombinant products. So, meron ding Bacillus subtilis, ganon. So, during my master's, I worked with the Bacillus subtilis. So, I transformed or I introduced a new gene or a plasmid into a Bacillus subtilis or microorganisms for it to express such a particular gene or particular peptide for antimicrobial reasons, antimicrobial activity. Okay, so Bacillus subtilis could be, and many other microorganisms can be used also as hosts for this series of useful products. Ganun. So nowadays, the vaccines of COVID-19 is an example of a recombinant DNA technology product. So they are cloned, right? So before introducing it into the viruses, they had to clone it into a particular host cell, and then it will then be introduced into the viral vector. Ganun. So there is cloning that happened with animal. So it uses recombinant DNA technology. So yun, I'm still ano nga ba yun? Looking at the internet, how did they massively produce these COVID-19 vaccines? Because it's quite different from the other vaccine production. Because of course, they now for the Pfizer and Moderna, they have to use mRNA technology. So I was still wondering, so I have to discover it more. I have to look at it in the internet more. So yun ang mga examples of the recombinant products. Okay, so the important factors in the design of these processes consist of one is the secretion of the product. It should be that there is um, there is an enough the proteins or the genes that are introduced into the host cell should be secreted out of the product. So, mo na siyang expression of the gene. Kitawag na siyang expression of this gene so that you will really have that protein product, a recombinant product. And then, second is the control of the onset of the synthesis during the fermentation. Of course, you have to know also the optimum condition of these host cells in order for the transgene, in order for the gene, the foreign gene, to be expressed out of the medium. Because it could be that your genes can be intracellularly produced or it can be extracellularly produced. So there are many kinds of enzymes or microbial products that that the microorganisms can excretely produce into the medium, while others they just have to produce it intracellularly. So we will tackle more about that later on, and then also in the InBt122 subject na. So it's in the downstream processing part. So while here, so different conditions such as the temperature, the pH, the agitation, the incubation period, and also, of course, it entails lots of optimization process. So it entails experimental experiments, use so many experimental conductivity for it to have a reliable conclusion. What are these optimum conditions for the host cell? So, and then the third is the maximizing the expression of the pharyngene. Of course, you have to see to it that the pharyngene is stable, because there are many cases, and it's really a natural way of the microorganisms that on the next generation, so the gene of the foreign gene will not be replicated on the another cells. Ganun yun. So when it is retransformed, kaya sa recombinant DNA process, there is a so-called selection marker or selection method. How will you know that these microorganism or these cells or these what they call that this colony has the foreign gene? By taking a look at it lang. So ganon. So you have to test these colonies, these cells, if they really possess the foreign gene. Of course, you have to extract the gene, and then you have to do PCR, ganon. So polymerase chain reaction, I think you know already that. So ganon class, yun ang sa recombinant DNA technology. And it is already 10:57 class, and I think that you have a class after this. So I think that I will just stop here. So just save your answers to the for next week na lang. Next week, because Wednesday ngayon, so Monday na po next week. So ayun, that's how these are the microbial products that are really significant in the industrial process. So I will continue next week with the chronological events of fermentation technology, and it has 4 waves: first wave, second wave, third wave, fourth wave. So there are many details to that. So I will continue for the another topic next week. So I will now stop share class, and then do you have any questions or clarifications regarding our topic today?
And so far, ma'am.
None? How about the others? Wala na?
None, ma'am.
Okay, so I think I will now dismiss you because it's already 11, so that you can prepare with your next class. Okay, so ayun. So thank you so much for joining the class, for joining this meeting
Thank you,
class.
ma'am.
Thank you,
Bye-bye.
ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
God bless.
Bye-bye.
God bless you too, ma'am.